Imagine a patient, right? They've got this, well, this massive infiltration of killer immune cells right inside their lung tumor.
Yeah, what we typically call a hot tumor.
Exactly, a hot tumor. By all traditional medical logic, the immune system is clearly recognizing the threat. The cavalry has arrived. And that patient should, respond beautifully to immunotherapy.
Right, they really should.
But they don't. The treatment just completely fails. The cancer progresses, and the clinician is left holding this biopsy report that says, everything was supposed to go right.
It's incredibly frustrating for everyone involved.
It really is. And today, we're looking at this wild diagnostic puzzle in oncology, where having the right cells, like all the right immune cells, doesn't matter at all if they're standing in the wrong place.
That paradox right there, having an army that is present but basically functionally useless, that is exactly why the research we're unpacking today is so critical. We're looking at a deeply fascinating paper from June 2026, published in Nature NPJ Precision Oncology. And the researchers essentially realized that to solve this life and death puzzle in non-small cell lung cancer, they had to totally abandon traditional cell counting.
Like throw it out the window.
Basically, yeah. Instead, they built this incredible artificial intelligence framework to track the actual, literal spatial social networks of millions of cells.
And that completely reframes our mission for this deep dive, because for you listening, we are going to decode exactly how these cellular neighborhoods are built, how AI interprets them, and why this shift moving from basic chemistry to literal tissue architecture is going to redefine precision medicine.
A massive paradigm shift.
It really is. And to visualize the leap we're making here, think about the old way of analyzing a tumor biopsy. It was essentially like taking a municipal census, right?
Right, just counting heads.
Exactly. You count the total number of protective units, say the immune cells, and you count the threats, the tumor cells. If the ratio looks good on paper, you assume the city is safe.
But it's just an average.
Yeah. But this new AI approach we're talking about, it's like tapping into the real-time GPS and like social media data of every single person in that city.
I love that analogy.
You aren't just looking at who is there. You are seeing exactly who's interacting with whom, who is isolated, and where the localized actual conflicts are happening on a block-by-block basis.
That GPS analogy captures the mechanical shift perfectly, because the actual terrain we're discussing, the tumor microenvironment or the TME, it is not just some static lump of tissue.
Right.
It's a highly dynamic, structurally chaotic ecosystem. I mean, you have malignant tumor cells, incredibly diverse populations of immune cells, stromal cells, vascular networks, and they are all arranged in these intricate, localized architectures.
But let me push back for a second, because if we know that, generally speaking, a high CD8 positive T cell density correlates with better survival, why isn't that enough? Like, if standard metrics, like the PD-L1 tumor proportion scores or just basic CD8 counts, if those are failing to predict patient outcomes consistently, it implies our tools are, assuming a homogeneous mix that doesn't actually exist, right?
Yes. That assumption of homogeneity is exactly the blind spot here. The TME is intensely heterogeneous. Its composition and its structure, they vary wildly from 1 millimeter to the exact next millimeter.
Wow, really? Just a millimeter away?
Oh, absolutely. So going back to your census analogy, if you have, say, 10,000 protective immune cells and 10,000 malignant cells in a big tissue sample, a basic density metric just averages that out.
It just says, hey, the forces are balanced.
Exactly. But what if all those immune cells are sequestered away in the stroma on the far north edge of the tissue, completely segregated from the rapidly dividing tumor cells in the center?
Ah, so they are technically in the same zip code, but they aren't actually engaging.
Right. The proximity is a total illusion. It's an illusion created by the blender of bulk analysis.
The blender, that's a great way to put it.
Density-based metrics completely strip away the spatial context. They ignore the architecture. And because they fail to capture those higher-order direct cellular interactions, patient responses to immunotherapy have remained, well, frustratingly unpredictable.
Which is terrifying for a patient.
It is. So the researchers recognized that to actually predict survival, we desperately needed a mechanism that could read the tissue not as a soup of cells, but as an interconnected network.
So let's do a really quick recap for everyone listening. Point 1, the tumor microenvironment is this chaotic, complex ecosystem that dictates survival. Point 2, Traditional cell counting is basically tossing the tissue in a blender and completely missing the actual spatial relationships.
Caught on.
And point three, to fix this, we need a tool that can read high-order cellular neighborhoods. Which brings us to the AI.
Enter the graph neural network.
Right, because to process 6.9 million cells across 506 lung cancer patients, the AI can't just be looking at a standard old-school stained biopsy slide.
No, absolutely not.
They must be tagging these cells with specific functional markers to feed them into the network as distinct data points. How did they do that?
So they used multiplex immunofluorescence, or MIF.
Okay, MIF.
Yeah, and what MIF allows them to do is capture spatial data in situ. So they preserve the exact physical architecture of the tissue while simultaneously identifying the functional states of every single cell.
That's incredible. What markers were they actually looking for?
They tagged 5 specific markers, and this is crucial. First, they used cytokeratin, or CK, just to identify the tumor cells.
Okay, CK equals tumor cell. Got it.
Then CD8 for the effector T cells. Those are your killer immune cells.
The good guys.
Right. Then FOXP3, that locates the immunosuppressive regulatory T cells, or TREGs.
So those are the ones that kind of calm the immune system down, which in cancer is a bad thing.
Exactly. They suppress the good guys. And finally, and crucially, they tagged PD-L1 and PD-1.
Ah, the checkpoints.
Yes. They used those to map exactly where the immune checkpoint suppression was actively happening. PD-L1 is like an immunosuppressive shield that the tumor uses.
Okay, so they have this incredibly rich, spatially preserved data set with all these colorful glowing markers. But how does the AI actually translate that visual fluorescent image data into a mathematical model? What is the actual logic of a graph neural network?
That's the genius of it. A graph neural network, or GNN, is brilliant here because unlike a standard convolutional neural network, which just looks at pixels in an image, a GNN looks at topology.
Topology, like the study of geometric properties and spatial relations.
Exactly. It looks at relationships. The algorithm takes every single tagged cell and turns it into a mathematical node.
Okay, one cell equals one node.
Right. Then it draws edges, basically connecting lines between cells that share a physical boundary.
Wait, so they have to be physically touching?
Yes. If a CD8 positive cell's membrane is physically touching a tumor cell, the AI mathematically registers that edge as a functional relationship.
It's mapping a literal social network, but the currency of connection is physical contact.
That is the foundation. And from there, the algorithm constructs a unique neighborhood around every single center cell. It expands outward up to 4 hops.
4 hops. What does that mean in this context?
So hop 1 is direct physical contact, the cells touching the center cell.
Okay.
Hop 2 consists of the neighbors of those immediate neighbors, and so on, out to 4 degrees of separation. And on average, doing this created highly specific neighborhoods of about 42 cells.
Wow. So instead of asking, what is the overall density of this entire tumor, the GNN is basically asking, for this specific cell right here, what is the exact localized environment within a 42-cell radius?
Exactly.
And it uses that microarchitecture to predict a hazard ratio, essentially evaluating whether that specific spatial arrangement of 42 cells helps or hurts the patient's overall survival.
It does. And honestly, the predictive power of this topological approach is just staggering. On their test data set, the GNN achieved a concordance index, or C-index, of 0.82.
Okay, for context for the listener, a C-index of 0.5 is basically a coin toss. It's random chance.
Yep.
And hitting 0.82 in a system as chaotic as human biology is incredibly high. It means the model is remarkably accurate at ranking which patients will survive longer based purely on these cellular neighborhoods.
To really appreciate how good that 0.82 is, we have to look at the baselines they tested it against.
Oh, right. What were the alternatives?
Well, if you used CD8 positive density alone, so just counting the sheer presence of killer T cells, the C-index dropped to 0.75.
Okay, quite a bit lower.
But more importantly, if they used a network that only looked at the spatial arrangement but stripped away the marker data, so the AI knew the geometric shape of the crowd but didn't know who was who, it scored 0.715.
That is such a phenomenal data point. It proves you can't just know who is in the tissue, and you can't just know the structural shape of the tissue.
Right.
The predictive power relies entirely on knowing exactly who is standing next to whom. The identity and the topology are inextricably linked.
Completely linked. And the GNN's predictions were statistically independent of the patient's clinical tumor stage and independent of whether they had received prior immunotherapy.
Wait, really? It didn't matter what stage of cancer they officially had.
Nope. The localized spatial architecture of the cells tells the story of disease progression independent of our traditional macroscopic staging.
That represents a profound shift in how we approach cancer prognosis.
Right.
So let's do another quick sub-summary here. Point 1, the GNN turns tissue into a network graph, where nodes are cells and edges are physical contact. Yep. Point 2, it analyzes these 42 cell neighborhoods up to 4 hops away. And point 3, it is highly accurate at predicting survival, crushing traditional metrics independent of clinical stage.
That's the perfect summary.
But, concordance index of 0.82 is ultimately just a number, and AI is notoriously a black box.
Oh, absolutely.
It spits out this highly accurate hazard ratio, but it doesn't natively explain why that specific arrangement is hazardous. For a clinician to actually develop a targeted therapy based on this, we need to understand the biological logic driving the algorithm. How did the researchers extract the why from the GNN?
They had to peek into the AI's brain, basically, using a technique called latent space clustering.
Latent space. That sounds very sci-fi.
It does. Latent space is essentially a complex, multi-dimensional mathematical map where the AI plots all these millions of cellular neighborhoods.
Okay.
Neighborhoods that share similar underlying spatial dynamics and similar immunosuppressive patterns. They naturally gravitate toward each other in this space, even if those patterns aren't immediately obvious to human eyes.
So they're letting the AI tell them how it naturally categorizes the microenvironment.
Exactly. The researchers applied a statistical method called k-means clustering to this latent space, and they discovered that the AI had essentially grouped all the spatial neighborhoods into 7 distinct biological states.
7 states. Let's explore what those states actually look like, because I think this is where the biology gets genuinely fascinating for the listener.
It really does.
If we look at the extremes, cluster 5 is the optimal scenario, right?
Yes, cluster 5 is the heroes.
Right, the heroes. It features high densities of CD8 positive effector cells, high PD1, and critically, extremely low populations of tumor cells expressing the PDL1 cloaking protein.
Exactly. It's a highly active, totally unsuppressed immune response. And unsurprisingly, the AI strongly associated cluster 5 with favorable survival.
Okay, so that's the good one. But then you have the antagonistic extremes, right? The villains, clusters 4 and 6.
Yeah, the villains. Cluster 4 is what oncologists call an immune desert. It is dominated by tumor cells with almost no immune infiltration whatsoever. The immune system simply isn't there.
Wow. Just a desert.
Literally. And then cluster 6, however, is an immune evasion zone. So it is packed with both tumor cells and immune cells, but the tumor cells have heavily upregulated their PDL1 expression.
Meaning they've turned on their shields?
Exactly. They have deployed their immunosuppressive shields, effectively neutralizing the immune cells in their immediate physical vicinity. Both cluster 4 and cluster 6 strongly predict poor survival.
Okay, but the real revelation here, the cluster that completely breaks our old density models, is cluster 1.
Oh, cluster 1 is so interesting.
The researchers identified this state as a neutral survival predictor, which sounds kind of boring until you actually look at the composition.
Right?
Because cluster 1 contains massive amounts of killer CD8 positive T cells. Under conventional metrics, a pathologist will look at that, flag it as a highly inflamed hot tumor, and predict a great outcome.
They absolutely would.
But the GNN saw something else. It saw that cluster 1 also contains massive amounts of PDL1 positive tumor cells right alongside them.
It is the biological definition of a stalemate. You have immense immune pressure pushing against the tumor, but the tumor's localized evasion mechanisms are pushing back with equal force.
It's basically an ecological battleground. If you bring a massive army of CD8 cells, it doesn't guarantee a victory if the enemy has a powerful PDL1 shield generator physically adjacent to your front lines.
That's exactly it.
The AI recognized that stalemate and predicted a neutral tug-of-war outcome. Traditional density metrics, which just count the total number of CD8 cells without seeing the adjacent shields, would have gotten that prediction completely wrong.
To build on that, finding the stalemate state proves that simply classifying a tumor as hot or cold, based purely on inflammation, is a dangerously simplistic paradigm.
Dangerously simplistic.
Yes, because a hot tumor could be a productive tumor-killing environment like cluster 5, or it could be an exhausted, neutralized stalemate like cluster 1. The spatial context, specifically the physical proximity of suppressive markers like PDL1, is literally the only way to differentiate them.
So the key insight from peering into the latent space is that having a high immune presence is only functionally relevant if the localized suppressive markers are low. The AI successfully identified these complex tug-of-war zones, proving that immune density alone is a deeply flawed prognostic tool. So to recap this part for the audience, K-means clustering revealed 7 distinct microenvironment states. High immune presence only works if tumor-suppressive shields are down. And the AI found these biological stalemates that human pathologists were missing.
Which brings us to arguably the most sci-fi and potentially the most clinically actionable part of the entire paper.
Oh, this is my favorite part.
It's one thing for the AI to just observe and categorize these neighborhoods, right? It's another thing entirely to test the physical rules of this cellular world. The researchers utilize the GNN to perform in-silico manipulation.
A digital sandbox.
That's digital sandbox.
Because the GNN has mathematically mapped the exact relationships of the tissue, they can computationally alter it. They're essentially playing SimCity with the digital tumor.
That is exactly what they're doing.
They're artificially swapping the identities of cells in the latent space to see how the survival prediction shifts in real time.
The counterfactual power of this is just incredible. They asked the GNN, what happens to the hazard ratio if we digitally mutate a tumor cell into a CD8 positive effector T cell?
And as you would expect, adding killer T cells improved the survival prediction.
Right. But the critical finding wasn't that it improved, it was where the improvement occurred.
Yes, the spatial constraint. When they digitally injected the CD8 cell into the microenvironment, the survival prediction improved significantly more if that cell was placed in direct physical contact with the tumor cell.
Exactly.
If they placed the CD8 cell just hovering nearby in the stroma, separated by even a tiny gap, the predictive benefit dropped off drastically.
And that proves that localized direct cell-to-cell interaction is the primary driver of tumor suppression. But they didn't just test the immune effectors, they applied the exact same in-silico manipulation to the suppressive cells, and the results were, well, they were devastating. Let's look at the FOXP3 positive regulatory T cells.
The TREGs. And this fundamentally reframes a decade of conflicting medical literature, right? I mean, we've had major papers arguing that TREGs are a primary driver of poor prognosis, and then other studies showing they have a completely negligible impact on survival. It's been a massive source of debate.
It has been huge. Historically, the overall density of FOXP3 positive cells has shown incredibly noisy, mixed results in the literature. But when the researchers used their digital sandbox to physically force FOXP3 positive cells to touch the killer CD8 cells, all that noise just vanished. The patient's survival prediction plummeted.
Wow. And the more physical, direct edges they drew between the TREGs and the CD8 cells, the worse the prediction got.
Yep.
This suggests that all those conflicting studies were likely both right.
Mm-hmm.
They just lacked the spatial resolution to see if the TREGs were actively engaged in a physical interaction.
Precisely.
Having TREGs just hanging around in the stroma doesn't inherently doom the patient. It's about whether they have their hands around the throat of the effector cells.
Spatial arrangement physically governs immunosuppression. Direct contact is required for these suppressive cells to turn off the CD8 response. And they validated this mechanism beyond just the TREGs.
Oh, they did? With what?
They perform the same in silico manipulation with PD-L1 positive immune cells, which includes populations like tumor-associated macrophages. And once again, when they computationally force these macrophages to directly touch the CD8 cells, the survival predictions just cratered.
It's the physical chokehold. Altering the digital tissue proved unequivocally that effector cells work best in direct contact with tumors, and suppressive cells actively destroy survival odds only when they are in direct contact with the effectors. Physical proximity isn't just correlating with immune function, it is dictating the biological outcome.
Exactly. So what does this mean for the future of oncology? Because this isn't just an elegant computer science exercise.
Right, it has to mean something for the patient.
By decoding these exact spatial interactions, we are laying the groundwork for next-generation spatial biomarkers.
So how does a spatial biomarker change the clinical reality for a patient sitting in a consultation room? What's the practical application?
It explains exactly why monotherapies fail, and it dictates how we design combination therapies. If a patient's spatial biopsy reveals that their tumor is dominated by cluster 1 stalemates, or that their CD8 cells are caught in physical chokeholds by FOXP3 TREGs, administering standard PD-1 immunotherapy simply won't save them.
Because the effector cells are physically restrained.
Exactly. But if you read that spatial biomarker, you know you need to combine standard immunotherapy with a targeted agent that locally depletes TREGs or disrupts that specific macrophage interaction. You have to physically break up the bad neighborhood dynamic to allow the immune system to do its job.
We are officially transitioning from chemistry to architecture. The physical structure of the disease is the disease itself.
The topology is the pathology. When we understand the interactions at a microscopic topological level, we can start engineering highly specific interventions that disrupt the tumor's localized support networks, rather than just indiscriminately bombing the whole system.
That is just incredible. As we wrap up this deep dive, I want to leave you, the listener, with a concept to chew on. We started this discussion talking about medical imaging, simple, clean X-rays that show you what is broken.
Right.
And we've just explored how a graph neural network can accurately map the chaotic, multidimensional social networks of millions of cells to predict if someone will survive lung cancer, fundamentally outperforming our best density metrics. But take the logic of that in-silico sandbox one step further.
Okay.
If an AI can play SimCity with a digital tumor, artificially swapping cellular identities and drawing new edges to figure out exactly which physical handshakes are killing the patient, what happens when we start designing drugs, not just to kill cancer cells, but to actively rearrange their social networks? If we can digitally mutate the microenvironment to save a life in silico, how long until we develop therapeutics that can physically break up those specific cellular handshakes inside the human body? Think about that. The next time you hear a doctor or a researcher talk about the future of precision medicine, remember, the true precision isn't just knowing the enemy is present in the tissue, it's knowing exactly who they are standing next to.